Hello everyone, welcome back to my channel. In this video lecture, we will cover a new chapter of biology, that is photosynthesis in higher plants. So let's begin with video. Photosynthesis is the physiochemical process. Living forms on earth depend upon sunlight for energy. In these plant type, photosynthesis denoted by PTSN. Important of photosynthesis: primary source of food and O2 liberates. What do we know? Variegated leaf experiment, presence of sunlight. Second, half leaf experiment, CO2 is essential for photosynthesis. Then early experiments: Joseph Presley Bell, Charmin plant experiment, Jan Ingenhouse, Julius von Sachs, T.W. Engelmann, and Cornel van Niel. Then where does photosynthesis take place? In mesophyll, have large amount of chloroplasts arranged along walls get proper light. The membranous system are useful in trapping light and synthesize ATP and NDH, which comes under light reaction. In stroma, synthesis of sugar takes place, which comes under dark reaction, which depends upon the products of light reaction, but it doesn't occur in dark only. Then what is light reaction? The light reaction of photochemical phase includes light absorption, water splitting, oxygen release, and formation of ATP and NDH. The electron transport: electrons are excited from PS2 and transported to electron acceptor. It transfers electron to an electron transport system consisting of cytochromes, which is downhill movement. Then the electron transport done at inner side of the membrane of thylakoid to support the electron to PS2 and release oxygen. Chemosynthetic hypothesis: after splitting of water, H plus get accumulated in lumen of thylakoid. When electron moves through photosystem, the H plus travels from stroma to lumen. These three events creates a proton gradient between stroma and lumen. Then why are ATP and NDH used? Because they are used in biosynthetic phase, dark reaction to synthesize sugars. After World War II, Melvin Campbell concluded that first product of fixed CO2 is 3-phosphoglyceric acid or PGA by radioscopy and using radioactivity 14C. And after some time it was concluded that in some plants, first product was again an organic acid with 4C atom. Hence, based on two different first product, two pathways were introduced called C3 and C4 pathway. Kelvin cycle is the important interpreter of CO2 is 5C RUBP, and it was regenerated, found in all photosynthetic plants, whether C3 or C4. Then the C4 pathway is present in plants which are adapted to dry tropical region. Leaves of C4 plants have crans anatomy, that is bundle sheet surrounds vascular bundle. Crans means wreath and its reflection of the arrangement of the cells. They have many chloroplasts with tough walls impervious for gaseous exchange. Mesophyll lacks rubisco and OAA is formed in mesophyll. Then OA forms 3C malic acid or aspartic acid, which goes into bundle sheath and release CO2 to form C3 compounds, and that compound further sent to mesophyll. Photorespiration: rubisco is the most abundant enzyme. It has more affinity for CO2, but if O2 is to CO2, then for O2, hence the binding becomes competitive. Factors affecting photosynthesis: light, CO2 concentration, temperature, and water. These are the NCERT diagrams for your reference. This is a diagram or section of chloroplast. This is the light harvesting complex, cyclic phosphorylation, ATP synthesis, and diagrammatic representation of Hatch and Slack pathway. So this much for today's video. Hope you liked it. Don't forget to subscribe my channel and don't forget to like, share and comment my videos. Thank you.